There's 3 different sections. So when you go in here, see there is C. C has it.
You have to scroll, because like I've opened other sections before and been like, oh yeah, that's what we're talking about, and it's not the right.
Yeah,
thing.
B
Like,
goes
I
for...
forget what chapter this is off the top of my head because I'm just piddling on my computer, and then I'll pull it up and I'm like, yeah, you know, genes, and it's not the right gene.
Yeah, well, each of them are like 80 pages long, so you got to scroll way, way down. We're in 2 right now. Okay, so with the dominantly inherited disorders, achondroplasia is one of the forms of dwarfism. This one affects 1 in 25,000 people. And with this one, it's a little unusual because if they're heterozygous, the individuals will have the dwarf phenotype. So if they just have just one gene for the dwarfism, they will have the phenotype in there. So there isn't going to be any carriers of the dwarf. If the gene is there, you will be short in there. And so with dwarfism, though, there is multiple causes of dwarfism. This is just one of them in there. So the dominant alleles for lethal diseases are much less common than for recessive diseases in there because the lethal effects don't allow there to be any carriers, and a lot of times it's going to kill the person before they can reproduce. And that stops them from getting spread. An exception would be Huntington's disease, which is a late onset lethal disease. It's a degenerative disease of the nervous system in there, and the person has no phenotypic effects until they're 35 to 45 years old. So most of the time, if they're going to reproduce, they've done it by then. And it can be passed on. And this affects 1 in 10,000 in the US. They can do genetic testing for this in there if the person is aware that they are a carrier of this or that they have the allele for this. They can, or if they suspect they might have the allele for it, they can do genetic testing for it. Well, they may, if they only have it on one gene in there, they have a 50-50 chance of passing it on. But they don't necessarily know they have it at the time they're reproducing. Because you're usually not going to have any symptoms of it, any clue that you have it, till you're past the childbearing years. So some of the disorders are multifactorial in there. So there's a genetic component plus significant environmental influences in there. So, you know, there's some people who say, well, I don't worry about what I eat because there's no heart disease in my family. Well, yes, there is a genetic component in there, but there's also a huge environmental component in there. And so it's that way with diabetes, cancer, alcoholism, schizophrenia, bipolar disorders, lots of other disorders in there. So your genetics alone doesn't determine your fate in there. One really good example, if you have the breast cancer gene, you have a 20% increased risk of having breast cancer, not 100%. 20% greater chance than a normal person of having it. So what goes on in your environment can have a significant influence. No, you don't know that you have the gene in there. You would have to, basically, they would look at a pedigree of your family and decide, is there a possibility that you would have it, and then do the genetic testing to see if you have it. There is a chance that you have the gene. It depends if there's a chance for any of them, but you wouldn't know. You would have to look at the pedigree going back to see right now, because the pedigree could rule out and going down in there. Because when they look at the pedigree, they actually will go back on both sides of the family, and there are other cancers in there and other things that can be along with the gene in there that they'll look and say, okay, now we know this. So when they look to decide if they're going to do the test, they look to see, are you likely to have it in there? If you're not likely to have it, You can still get the test, but you'll be paying for it out of your own pocket in there. And even if you are likely to have it, that's no guarantee the insurance will pay for testing in there. It's just if you have a policy that does cover those things, that has to be done beforehand. And so they may actually, being that your sister has died from breast cancer, she may not have the gene for it either. So they want to look at those other things. So like when they looked for me, it was going to involve having to have one of my aunts needed to be tested first before they would consider covering it for me. It's about $5,000 for your first test. Stress can do that in there. With the environmental factors, we know they're environmental factors because the person can have the gene and not get the disease. We don't always know what all the environmental factors are. And it's one of those things that it's really hard to study that because it's not ethical to say, well, you know what, schizophrenia runs in your family, so we're just going to torment you and see what happens. It's not right to do that to people. So with the genetic testing and counseling in there are a lot of tests available out there. They come up with new ones all the time in there so that you can look and see and assess the risk prior to conceiving a child. Not everybody agrees with doing genetic testing in there. So, well, a person may look at it and say, well, what's the harm in doing the breast cancer testing in there? Maybe you don't want to know. Maybe you look at it and say, yeah, there is a chance that I have this gene and I don't want to know. I'm going to live my life and try and be healthy as I can, but I don't want that hanging over my head and causing the stress in there. You don't want to force those people to do it. But also in there it gets into ethical issues about some people don't believe that genetic testing should be done on embryos in there. And if they have a terminal disease in there, is it okay to terminate it? So it starts getting into all kinds of ethical issues where people have different views, and some people will look at it and say, well, it's your life, you do what you feel is right by your beliefs, leave me alone to make mine. But others say, no, nobody should be able to do that. So it just is one of those things where when they make laws for it, there's always going to be somebody who is way outside the bell curve on their thinking. So they almost have to make laws to prepare for that person who's going to be like, well, I think we should have you give birth to it, and we should just test that creature that you give birth to no end. It's going to be some really extreme thought in there. So it's one of those things that as a normal person, anything you look at and think of in there, there's always going to be somebody who's got twisted thoughts that have never come into your mind out there. So with fetal testing, amniocentesis can be done between weeks 14 and 16. They take out about 10 milliliters of amniotic fluid. Some of the disorders can be detected by the presence of certain molecules in the fluid. Others you can test in the DNA from some of the fetal cells that get sloughed off in the fluid. They can do a karyotype. There's some risk with any procedure. I mean, it's a big needle. I can't imagine that it feels very good putting it in. Never had it done, but I just don't think that it looks like it could be pleasant in there. Chorionic villi sampling in there. The tube's inserted through the cervix and suctions out a sample of the placenta. That can be done a lot earlier, the 8th to 10th week in there of pregnancy. The what? No. They just take off a few cells. Any procedure like that does run the risk of aborting it in there. It's always a risk. So usually these are done on older mothers. If you're over age 30 when you get pregnant, you're considered at increased risk for chromosomal abnormalities in there. Over 30. So, you know, it kind of looks at, most women can't even get pregnant over 40 in there. Well, it happens, but if you are 25 years old and you're like, I'm going to wait till I'm 40 to keep trying, but I really want a family, you're setting yourself up for heartbreak. Because you may not be able to. You don't know how long your fertility will last. 40 is getting pretty old to be having a baby. You have high risk of chromosomal abnormalities as a female. And what are you going to do? Go get you're going to be a cougar and get a young guy in there? Or you're going to get some other guy that's close to your age in there, and then you've got increased risk of mental disorders in there. Your child may never leave your house in there if you start too late. Well, I mean, it's just there's definite risks with being older in there. It starts increasing at 30, and at 35, it's a logarithmic curl. Yeah. Well, they don't nobody's going to make you do them. Yeah. It's really unlikely that they're going to hurt the baby. They can guide things with the ultrasound. It's more that you're introducing a foreign object in, potential risk of infection and trauma to it. They're not going to hit the baby with it in there because they don't go in that deep with it. And even with the chorionic villi in there, I mean, yes, there is a hole in the cervix. However, if you've ever had them insert anything in through the hole in the cervix, it's not a hole that likes to have things put through it in there. It just, you know, it's not good, feeling good when the baby comes out. It's not going to feel good when you go the other way through it in there. It's going to, you know, the uterus isn't going to be happy. It doesn't want to be poked with something sharp. So ultrasound, this is non-invasive in there. It allows for screening of anatomical abnormalities in there. So with that, though, the fetus actually has to be a significant size that you could see the abnormalities in there, and not all abnormalities are going to be visually observable. With blood tests, some of them can now isolate fetal cells and some of the DNA that's escaped into the mother's blood. You usually are going to only get very few cells in there, but they may have to be cultured to be tested in there. Then they can screen newborns as well. So with PKU in there, the phenylketonuria in there, they can't process phenylalanine, and toxic byproducts would accumulate with that can cause mental damage. And so if they know early on, the manifestation of the disease can be much milder because they can treat it with a special diet and avoid certain things in there. So one of the things that they cannot have is NutraSweet or aspartame. So you may see warnings on certain things, you know, warnings set for, you know, for PKUs in there with it. So with the chromosomal basis of inheritance in here, we're on chapter 15 now. So what this is basically operating on is the idea that the Mendelian genes have a specific location along the chromosome, and when the chromosome undergoes segregation and independent assortment, the gene that's with it's going to go there with it in there. And so this picture is just showing the segregation of the chromosomes, and you've got the gene on the chromosomes in there that follows it. So the law of segregation says that the two alleles for each gene separate during gamete formation, and independent assortment says those alleles on non-homologous chromosomes are going to assort independently. Oh, where did we have those?
Right there in the top.
Oh, okay. I don't know if those are Rowena's for something. Were the other little medicine cups over there at the other building?
No.
Okay.
Nothing over
All right. We have two. They'll have to share.
there. Okay, but there's like about, there's a few dozen of these.
Yeah, I just don't know what, I didn't order them and they're not mine, so I don't know what she uses them for. Okay. I just don't want to put her in the position of thinking she's got supplies for something and she comes in and is like, oh, they're gone. So if you have the genes on homologous chromosomes in there, they're not going to sort independently. If it's on the same chromosome, usually the two genes have a better chance of traveling together in there. So the person who did a lot of research in this field was Thomas Morgan in there. He was an experimental embryologist in there. And he was initially skeptical of Mendelism and the chromosome theory. So what he ended up studying was the Drosophila melanogaster fruit fly in there. It's a fruit fly. I mean, how can there be anything nice about it? But the reason he used the fruit flies was, like the peas in there, prolific breeders. Nobody cares if you inbreed a fruit fly in there. They have a new generation every 2 weeks. So if you feel like if you've ever gotten fruit flies stuck in your house and you're like, they just keep coming, they do, literally every 2 weeks in there. They only have 4 pairs of chromosomes, 3 autosomes, 1 sex chromosome, so it makes them easier to study. The wild type is going to be the phenotype character that you see most commonly in the natural population, where the mutant in there is the alternative phenotype that you would see in there. And so it's assumed to be a mutation from the wild type. So if we look at things that way, having lactose intolerance is actually a wild type phenotype. And those of you who drink milk in your later years without it giving you gas, which most of the time it does, you just don't realize it's abnormal amount of gas, you're mutants. So what he studied was eye color on these fruit flies. And a lot of the labs that they have you do in most books call for fruit flies. I think your book even has one for fruit flies. You get the same results with the fruit fly that you got with the peas on here. So we're not going to use fruit flies because once a fruit fly is in some place, they are really hard to get rid of. And I can't guarantee that you guys wouldn't let one out and then I would be plagued for the rest of the years here. So it's just showing where if you cross one with red eyes and white eyes in there, the red is the dominant trait. All the offspring in the F1 generation will have FY or red eyes. In the F2 generation in there, you're going to get 3 with red eyes, 1 with white eyes in there. And that's the same ratios you got with the peas in there. So it's just showing the same exact thing. It's just showing the gene being on the chromosome here in the picture is the only difference. So the chromosomal basis of sex, your chromosomal sex is determined by your chromosome in there. So for females, you get 2 X chromosomes. For males, it's an X and a Y chromosome. You're going to inherit 1 from each parent in there. So the Y chromosome in there only has small regions that are homologous to the X chromosome in there. The Y chromosome is a lot smaller, 78 genes on there, the Y chromosome about 1,100 genes. So it can't be completely homologous in there. So in humans, the signs of your sex physically begin to appear at about 2 months. So that's why they can't determine the sex of the baby really before that without... We all start out the same in there. So prior to 2 months, the gonadal development is generic in there. And you actually don't get the signal to start turning into a male until about 5 or 6 weeks. And that's the internal signal to turn on the other regulatory proteins in there. But physically on the outside, you're not even going to start to look like a male till 20 weeks. So halfway through, if you looked at an early ultrasound of yourself, that might not be a picture you want to show future girlfriends or something in there because you will be built like a girl in that ultrasound in there. Well, it's not very big.
Oh, I know.
I mean, tiny little thing in there that they're looking for. So what's going to actually determine what sex develops in there is one specific gene, the SRY gene. The sex determining region of Y is what SRY stands for. And that's going to code the regulatory proteins for the other genes in there. So if your SRY gene is not active in there or it's defective, you'll just continue developing as a female even if you have a Y chromosome. Occasionally it will cross over and it ends up on a Y chromosome. So about 1 in 10,000 normally appearing males are actually chromosomally a female, and about 1 in 10,000 normally appearing females are chromosomally a male. I wouldn't believe anything that was put on a Hollywood thing, though. So, with the Y chromosome, it's got 78 genes, 25 proteins in there. Some of them are duplicates in there. So a sex-linked gene is going to be a gene that's located on either of the sex chromosomes in there. So a Y-linked gene is going to be on the Y chromosome. This will be passed from fathers to sons. So why can't a female get that? Because you don't have a Y chromosome in there. So X-linked genes, there's about 1,100 of them on the X chromosome. They'll have a different pattern of inheritance since males and females inherit different numbers of X chromosomes in there. So when we do Punnett squares for sex-linked genes, what you've got here is you've got eggs, X and X, X and Y. You use a superscript instead of having it be the full letter like you do with the normal dominant recessive inheritance in there. So where you had the incomplete dominance, you had a superscript. So you had the letter that represented the trait and then the superscript that represented the allele. This is representing the X and Y chromosome, and up here is the allele up at the top in there. So that's how they're done. So when you bring these down into the boxes, you actually should have 4 letters in there so that you can see which sex is going to get which pattern in there. So X-linked traits are going to be passed, fathers pass them on to all of their daughters and none of their sons, because if they pass an X chromosome on to you, you're going to be a girl. Mom only gives you X chromosomes. So X-linked recessive traits are only expressed in females if they're homozygous. So they would have to inherit it from both mom and dad, where males only have to inherit it from the dad only on there. So color blindness is one that's almost always inherited as an X-linked disorder. Yes, you can have both males and females be colorblind, but it's much more common in males because you would have to inherit it from both mom and dad on there. So Duchenne's muscular dystrophy, it's X-linked, 1 in 3,500 males in the U.S. It causes progressive weakening of muscles and loss of coordination. These rarely live past their 20s. They're missing the dystrophin protein in there. But because it's X-linked, women will have it in there, be a carrier for it in there, and pass it on. It isn't that these males are necessarily passing it on. A lot of times they don't live long enough to pass it on. So hemophilia is X-linked recessive in there, missing 1 or more proteins for blood clotting in there, so more common to be in males. So a female ends up with 2 of these X chromosomes in there, but most of 1 of them are going to be inactivated during embryonic development in there. So the X chromosome's a little bit different in that you're going to have one or the other of them be active in there. The other one is going to become inactive as a bar body in there. And it's completely random and independent for each embryonic cell in there. And then all of its mitotic descendants will have the same X become inactivated in there. So the X inactive specific transcript is going to become active only on the chromosome that ends up becoming the bar body. So as a female, you may have on some of your cells the X chromosome from mom, and on some of them the X chromosome from dad. You can have a mix in there. So the link genes, they tend to be inherited together because they are near each other on the same chromosome in there. And so when you have crossing over, the closer they are together, the more likely they are to move together on there. So basically the linkage is traits are usually inherited in specific parental combinations because genes for characters near each other on the same chromosome are more likely to go in there. So when we look at the recombination of unlinked genes, you've got the independent assortment in there. So you'll have the parental types, which are the phenotypes of the parents, and the recombinants or the recombinant types are the new combination of phenotypes in the offspring. So if you have a 50% frequency of recombination for any two genes, that's considered normal because you have a 50-50 chance of getting one from mom or one from dad in there. When you start having more than 50%, that's when they are linked in there. And the more above 50%, the stronger the linkage is in there. So with crossing over, it accounts for the recombination of linked genes in there. And so, let's see, this picture here. It's really hard to see up on the board. I know it's small up here. But basically it's showing where you've got like this B gene and this VG gene in here. And then it's showing it over here, how they're in two different places. If you have crossing over of this section, they can split. But in here, you don't have crossing over with this particular one. So it's going to be a greater than 50% chance because they're next to each other in there. Yes, it can happen that they cross over, but most of the time they're not going to cross over and end up separately in there. So we can do where you calculate the recombination frequency. It can be calculated. You're not going to do it in here. But you can build genetic maps doing this, and there's different types of genetic maps that can be made, but they look at an ordered list of the genetic loci along a particular chromosome. So they're looking at, okay, what genes do we have and in what order are they on the chromosome in there? So one of the things they will look at in trying to determine what order they're in is the recombination frequency in there. And so the higher, basically it proposes that the further apart two genes are, the higher the probability that a crossing over will occur between them, and you'll have a higher recombination frequency in there. Linkage maps then are based on the recombination frequency in there, and the map unit defines 1 unit is equivalent to 1% recombination in there. So what you want to know about that is basically it's a map based on how often things recombine in there. The cytogenic maps are going to locate genes with respect to the chromosomal features in there. So we usually find when we look at these different types of maps, the linear order of the genes is the same, but the spacing between them will vary in there. So we don't have really a good way of spacing them determining accurate spaces in there, and we don't even know, are the spaces necessarily going to be exactly the same all the time. So when you have alterations of the chromosome numbers in there, large-scale chromosomal alterations in humans and mammals usually are not going to survive. Most of the time they'll just spontaneously abort. And those who do survive are going to have developmental disorders in there. So how this occurs is with a non-disjunction. So in meiosis in there, meiosis 1, you've got where these things should be coming apart like this. If they don't, what you end up with is this in meiosis 2 instead. It doesn't necessarily mean it stops the process, but then what you get here are some of them having 3 of those chromosomes on there, some only having 1. They're supposed to have 2. If it occurs in meiosis 2, you do have a chance of getting some normal ones in there, but you also have a chance of getting 3 or 1 in there. So the earlier on the non-disjunction occurs, the greater your odds are of having an abnormal chromosome number in there. And so what they see with the chromosomal disorders is, as a female, the older you get, the more of this tends to happen in there. So there's definite higher correlation between age of the mother and the chromosomal disorders. You do see a little bit with the father in there too. So aneuploidy is when your zygote has an abnormal number of a particular chromosomes. Monosomic is when it has no copy of a particular chromosome, leading to one missing in there. Trisomy is when you have an extra one in the zygote. Polyploidy is when you have more than 2 complete chromosome sets in there. Plants will have that occur quite a bit, but not in animals in there. So those are changes to a whole entire chromosome. You can also have changes in just a gene in there. Just want to see where we are on things. So a deletion is when a chromosomal fragment is lost or deleted. You can actually have even an individual base pair in the DNA be lost or deleted in there. But with the chromosome, when it happens in there, it falls off, and a lot of times it ends up attaching to the sister chromatid in there. So you have a chunk fall off of one and it ends up on the other. So one chromosome has too much stuff, one doesn't have enough in there. So the duplication is going to be the deleted fragment that ends up on the sister chromatid in there. Inversion, that's where a chromosomal fragment reattaches to the original chromosome in the reverse orientation. So there is one substance we know that triggers that to happen. Anybody have any idea what it is? So asbestos, we know, triggers that kind of a mutation to happen. And then translocation, that's going to be where a fragment joins a non-homologous chromosome in there. So it comes off and it ends up somewhere else, and it's not necessarily on the sister chromatid in there. So a lot of the large deletions in the X chromosome in a male will be lethal in there. The duplications and translocations will tend to be harmful in there. So some of the disorders in here from chromosomal alterations, Down syndrome or trisomy 21, it's 1 in 700 children born in the U.S. that get an extra 21st chromosome in there. So they do have characteristic facial features, will tend to be short. They'll have heart defects that a lot of times can be corrected in there. Developmental delays. They have an increased chance of getting leukemia and Alzheimer's. So a lot of them will develop Alzheimer's by the time they're in their 30s. And their decreased risk, or they do have a decreased risk of high blood pressure and atherosclerosis. And the increased frequency of this happening for an infant increases with the maternal age. Most of the time it's due to a non-disjunction during meiosis 1 in there. So aneuploidy of sex chromosomes, these appear to have less effect than on the autosomes. The Y chromosome has relatively few genes, and so you just don't have as big of an effect there because not as many genes are affected. And then with the X chromosome, if you get extra ones, a lot of times they get inactivated as bar bodies in the somatic cells, so the effects can be reduced because of that. Klinefelters, this is an extra X chromosome, XXY, 1 in 500 to 1,000 live births will have this. They'll have male sex organs in there. So they have small testes, they're sterile, an extra X chromosome that's inactivated. Some will have breast enlargements and female body characteristics in there. Most have some normal intelligence. XYY, this one is sometimes called the super male in there. It's 1 in 1,000 males. They have normal sexual development in there. There's no well-defined syndrome with it. It says that they're taller than average. There were some studies that were put out that said they would have higher testosterone levels and that there was more of a tendency for violence. And I went looking for some of these, and what I was able to find were the studies that critiqued it, and what they said is, well, they were more likely to beat their spouse. That doesn't mean they're violent. And I was like, okay. So you could take that to mean what you want in there. Yeah. But I haven't been able to actually find the studies on it to see in there. But to me, that is not a critique. So XXX, 1 in 1,000 females, usually healthy, no unusual features, taller than average in there, increased risk for learning disabilities, but they're fertile. Turner syndrome, that's monosomy X in there, 1 in 2,500 female births. It's the only known viable monosomy in humans. Phenotypically, they're female, but they're sterile. Sex organs don't mature in there. Girls with Turner syndrome can develop secondary sex characteristics if they're given estrogen therapy and will tend to have normal intelligence in there. And again, there isn't all that many of these that actually get studied in there. It kind of depends on the parent being, aware and saying, all right, I have a daughter here who is not going through puberty, and the parent actually doing something and addressing it in there. And there are some parents that have little issues in there. We're like, well, I don't want my little girl to grow up. And time frames are kind of critical in there where she won't necessarily have the opportunity to go through puberty and all the other normal stages in there. No, because they don't have the right number of chromosomes to pass on. So some structurally altered chromosomes, chat in there. This has a specific deletion on chromosome 5. They're severely intellectually disabled, small head, unusual facial features. And what stands out about this one is they have an unusual cry that sounds like a distressed cat in there. And these usually die as infants or young children. In there, the chronic myelogenous leukemia, there is chromosomal translocation that are implicated with some of those cancers in there, where you'll have reciprocal translation during mitosis of the cell that becomes a white blood cell in there, and you end up with a large portion of chromosome 22 being damaged in there, and it produces what they call the Philadelphia chromosome in there, which is a shortened chromosome 22. And it leads to uncontrolled cell cycle progression. So some other inheritance patterns. The genomic imprinting is something that's actually been kind of put out in the media quite a bit lately in there, where you have variation in phenotype depending on whether the allele is inherited from the male or female parent in there. In a lot of cases, it's just adding a methyl onto one of the cytosines of one of the alleles in there is enough to make a change to it in there. So it would occur during gamete formation, and it will end up silencing a particular allele on certain genes because they're imprinted differently on the sperm and the egg in there. And then the zygote only expresses one allele of the imprinted gene that's inherited from the male or female parent in there. So old imprints get erased in each generation in there, so the new gametes will imprint according to the sex of the person producing the gametes. And it's thought to only affect a small fraction of the mammal genome in there. Most of the known imprinted genes are critical for embryological development. These have been implicated in possibly looking at a genetic reason for homosexuality in there, that you actually end up with the gene being imprinted wrong from the parent in there. So it could be a genetic cause of it in there. The what? Yeah. So that's basically what it's saying, is that, you know, it is, that there is a genetic, that this is a possible genetic explanation for that in there. And so there's been other things in there where they've looked at kind of behavior patterns in there. When we look, you know, you can't just, there are certain things where you look and you watch little kids and little boys and little girls sometimes are very different in how they do things, and we don't actually see a different gene on the X or Y chromosome that would account for those behaviors in there. So they think genomic imprinting can kind of explain some of those behaviors in there. So inheritance of organelle genes. So you do have some genetic material that is outside the nucleus. So in plants, it's your chloroplasts and plastids. In us, it's in the mitochondria. Your mitochondria has a small circular DNA. The organelles reproduce themselves and transmit the genes to the daughter organelles, and they don't display Mendelian inheritance in here. Most mammals, including us, the zygote receives its cytoplasm from the egg, so your mitochondrial DNA is inherited from the mother's side in there. So you're just kind of, you're genetically a tiny bit more your mom in there because of that. Well, you know, in my family, we were kind of looking, and my grandpa was a bit on the creepy side, and my cousins were like, well, you're the ones that have the same last name. And I said, yes, but you have the mitochondrial DNA. You're going to be the old pervert in there. And he was like, ah. And I said, yeah, I can change my name. So with the mitochondrial genes in there, they make up protein complexes of your electron transport chain and ATP synthase. So they're going to basically affect how much ATP you can make. And some of your muscle and nervous system diseases are susceptible to that in there. Basically, there's a myocardiomyopathy that causes muscle weakness in there that is attributed to that. Leber's hereditary optic neuropathy in there that can produce sudden blindness in young people in there. That is in there. The mitochondrial mutations can also contribute, at least in some part, to diabetes, heart disease, Alzheimer's in there. So with some of those things, you might have a little bit stronger genetic tie in there from your mom's side of the family. So if I show up here one day and I don't know who I am, the Alzheimer's comes from my mom's side of the family, and we know who to blame. So let's go ahead and take a few minutes as a break in there. Let's go ahead and keep going with this. So the molecular basis of inheritance, here's where we start looking at the structure of the DNA. in here. So in 1953, Watson and Crick established the double helix model of DNA. So when you think about it, it really wasn't all that long ago in there that they even realized you had this twisty thing in there. We didn't know what the DNA did at that point. So then Morgan's group showed that the genes were part of the chromosomes, and they were trying to figure out, okay, so this genetic material here, what is this genetic material? And at that point, the DNA became the candidate of looking and saying, okay, we think this is the genetic material. So our hereditary stuff and figuring out how is this stuff passed on is really relatively new knowledge out there. And so for some of you that look and go, oh my God, I took biology in high school and I didn't learn any of this, a lot of this stuff is new. I didn't learn this stuff in high school. So it's, you know, it's just a field that is changing and vast amounts of new information are out there. So how they came about to understand this was a lot of it was through bacterial transformation. And transformation is a change in the genotype and phenotype due to assimilating external DNA in there. And they studied this in bacteria in there. There was evidence that the DNA could transform the bacteria and that viral DNA could program cells. And so they did some studying of bacterial phages, or phages, which are viruses that infect bacteria. All a virus is DNA or RNA enclosed by a protein coat. It's got nothing else in there. It's basically DNA, a shell around it, that's it. So for a virus to reproduce, it has to infect a cell and take over the host cell machinery in there. So they studied the T2 phage, and a lot of these phages got their names in there in the order they were discovered. So this was number 2 that infected E. coli in there, and they used radioactive proteins and found that when the phage DNA entered the cell, the radioactive proteins remained outside. So that told them that all went in was the DNA from there. So additional evidence that showed that the DNA was the genetic material, Erwin Chargaff in there, he studied the ratio of nucleotide bases and found that the number of A's is approximately equal to the number of T's, and the number of C's is approximately equal to the number of G's. And the base composition varies between species, but within a species, you're going to see the number of A's and T's be equal and the number of G's and C's be equal. So if you have 10 A's, you're going to have 10 T's to go with it in there. If you have 10 or 15 G's, you'll have 15 C's to go with it. Because A pairs up with T, G pairs up with C in there. And together they make up 100%. So when we look at building the structural model of DNA, it's a double helix shape, and they used X-ray crystallography to come up with this. So it's like a ladder where you've got the rungs across and you twist that ladder in there. So the helix makes a full turn every 3.4 nanometers. So there are 10 base pairs per turn, meaning they are stacked 0.34 nanometers apart. And the nitrogenous bases are paired up, one purine and one pyrimidine in there, so they're complementary. So remember, your purines are C, T, and U, the pyrimidines A and G. So A and T pair together with 2 hydrogen bonds. G and C pair together with 3 hydrogen bonds. And
I remembered it backwards then, so I thought it was a purine with
I'll double check on that, because that's kind of what I'm thinking too. Yeah, that might be an error in here. Purines are A and G, and the pyrimidines are. So, yes, that's the original way in there is correct. So you'll want to change this on your printout. Yeah, it's. Yeah, that just. Well, what I do when there's something like that, and I know it's different, is I highlight it like this so that when I come into it next quarter, I can tell people on the notes. However, when they switch my computers or decide we can't save anything, I get to start all over every quarter on that. I'm getting real good at reinventing my wheels around here. Okay, so when we look at the DNA replication and repair, it's going to be a semi-conservative model. So you've got this double helix, you've got these two base pairs that are sticking out with each other in here, and what's going to happen is they're going to separate, and then you're going to fill in the other complementary bases that will go with it in there. So the parental molecule has two complementary strands in there. When you separate the two strands, that it serves as a template that fills in the other one. And that's what's meant by being semi-conservative in there. So when you get started with this process, you've got your DNA that's double-stranded together, and you're going to have it separate in there. And the area where it starts to split apart in there is going to create an origin of replication in there. And it's just a sequence where you have the openings come up. Yeah. So you create this little bubble in there, and when the replication occurs, so in a bacterial one, it's double-stranded and it's circular. It's going to go both directions around the circle, and so you end up with it creating two new circles in there. In the linear model, you can have multiple origins of replications in there, multiple bubbles, and it starts, and the bubbles spread out till they eventually come together in there. So the replication fork is the Y-shaped region at the bubble. So this is a fork here, a fork here, a fork here on there. So helicase is going to be the enzyme that's going to help to untwist the double helix in there. And you've got a single strand binding protein that's going to pair the bind to the unpaired DNA strands to keep them from repairing. So it's kind of like if you have yarn that's all twisted together and you've got the different threads in there and you try and pull it apart, it's going to end up with all this tension being twisted at the other end, and when you let go, it wants to coil back up normally in there. The DNA would want to do the same thing, and so the helicase helps to unwind it ahead of you in there. So it's kind of like having somebody start to untwist the yarn a little bit before you do that pulling in there. And the single strand binding protein helps to hold it open. The topoisomerase is going to help to relieve the strain ahead of the replication fork. So that would be having another person way up the line, untwisting it in there so that you don't end up with it just being all coiled up and knotted up at the other end. I don't have any yarn with me other than what's in this. So the enzymes that are going to synthesize the DNA, they can't initiate the process. The enzymes can only add on the DNA, so you've got to have other enzymes that are going to initiate the process. So a primer is an RNA chain of initial nucleotides that gets produced during DNA synthesis so that you can get it started. The primase synthesizes the primer in there. So remember, ASE endings usually go with enzymes in there. And it's going to start a complementary RNA chain from a single RNA strand in there. It uses the parental strand as a template, and it's usually 5 to 10 nucleotides long. So the DNA strand starts from the 3' end of the primer. So you kind of look at it, and you've got 5', 3' end. That's a primer. And then you've got a 5' to 3' end of the DNA. So everything has got a left and a right. This is the left and right of the primer. This is the left and the right of the DNA. So when you synthesize a new strand, a DNA polymerase is going to be the enzyme that is going to catalyze the synthesis of new DNA. So if you think about it, polymerase means to make a polymer of something, and you're making DNA, so you're polymerizing DNA. That's what a DNA polymerase does. Eukaryotes have at least 11 different DNA polymerases in there. And here we're going to call them DNA polymerase. In your notes it will give some of the different numbers of them, but I'm not worried about you specifying the number. I want you to know, is it a DNA polymerase or an RNA polymerase in there? So most DNA polymerases requires a primer and a DNA template strand in there. So DNA polymerase adds nucleotides to the primer at a rate of about 500 nucleotides per second in bacteria and about 50 per second in human cells. So that's still pretty fast if you think about it. That's like adding on 50 letters per second in there, gluing 50 things together per second. And then in bacteria it's really fast, but bacteria can, their whole organism can double pretty quickly too. So each nucleotide is going to have a nucleoside triphosphate in there, so the monomer joins onto the growing end and the two phosphates are lost in an exergonic reaction, so heat will be released, and that's what's going to drive the polymerization. So being that the DNA strands are double-stranded in there, you're going to have anti-parallel elongation in there. So the DNA polymerase can only add to the 3' end of the primer. So if it can only add on to this end of things, remember you've got 5' to 3' going the other way. So you've got two strands running two different directions, like this. So if you're thinking elbow to hand goes this way on this strand, elbow to hand goes this way on this strand. But they're sitting like this. So when you split it apart like this in here, so it's split open in here, and you're moving along. The leading strand is going to be the one that's able to go 5' to 3' without any breaks in it. So as it's going along here, you just keep adding on in this direction. But as you expose this one, 5' to 3' goes this way, but you're exposing this way. So what you have to do is jump back and go this way a little. Jump back and go this way a little. Jump back and go this way a little so that you can read. So it's kind of like saying I'm going to read a book backwards. So you start at the top of the page and read back, then you go back a page, start at the page and read down and go back a page in there, and then piece it all together in your brain. So you've got the leading strand that reads like a normal person, the lagging strand that reads the weird format in there. But being that you can only add on to the 3' end, you have to go that way. So what it ends up making are these little pieces here that will have to get joined together. And these little pieces are called Okazaki fragments. Some Japanese person. Mr. Okazaki. That's what I should have named my dog. He needed a Japanese name. So these segments can be 1,000 to 2,000 nucleotides long in E. coli and 1 to 200 nucleotides long in eukaryotes. So shorter in eukaryotes. So they have to be glued together, and a DNA ligase is going to glue them together in there. So ligase means you're going to ligate or glue things together. DNA ligase, because it's DNA that you're gluing together in there. So is there any questions on that one? That's going to be one of those. I believe there is video clips on there of this that are really helpful to watch it in action. So when you look at it here, you've got this stuff in here, 5' to 3' in here. And it's giving the section numbers, and it's got DNA polymerase 3, DNA polymerase 1. Just know it's DNA polymerase. That's enough for now in there. So the DNA replication complex is the various proteins that participate in it and the DNA in there. So it makes a DNA replicating machine in there. And so the replication complex may not actually move along the DNA. It's thought that it pulls the DNA through the machine. So you're reading this in there, and when you're reading things, mistakes happen. So the errors in completed DNA are only 1 in 10 to the 10th in there. Your initial pairing, you're going to have 1 in 10 to the 5th nucleotides have a mistake in there. So as it goes along, the DNA polymerase proofreads it. Occasionally, it misses errors when it proofreads it. So one of the ways that it can repair things is mismatch repair. The enzyme comes along and it sees there's a mistake in there, so it removes it and replaces it in there. Other times you can have mistakes occur after it's already made in there and it gets mutated in there. The DNA is constantly exposed to mutagens, so toxic things in the environment that will do this in there. And some of them can cause permanent mutations that get passed on to successive generations. So in E. coli, we know there's almost 100 repair mechanisms. In humans, so far, we've identified 130 of them. There's probably a whole lot more out there that we just haven't identified. So the nice thing about there being so many is, well, more mistakes get created in you, but also when you start getting numbers like this, they no longer make you memorize them in class. That's too many. So you just go, yeah, there's repair mechanisms. They might give you a couple examples of how they work, but then it becomes something that, okay, well, if you specialize in that, then you'll memorize them. So nuclease is a DNA cutting enzyme, and it's going to cut out damaged DNA. And when it does, it makes a little gap in there that gets filled in with nucleotides. So the nucleotide excision repair is going to involve filling in that gap in DNA. Another type of mistake you can make is the Thymine Dimer in there, and it's not necessarily a mistake, but it's a mutation type that you can have. What will cause Thymine Dimers is UV light damage will cause this. So it ends up covalently linking the thymine bases that are adjacent to each other on the strand, and then they're no longer read as thymines, and you end up with it being a GC pair instead of an AT pair. So the evolutionary significance of these mistakes, occasionally some of the mistakes make it through and they enter into the gene pool that creates more diversity in the species in there. There is a small portion of the cells that has the DNA on there that the DNA polymerase cannot repair. Since you can only add to the 3' end, where you start here, this base is at the 5' end. There is no 3' until down here. You can't actually fix the very end ones in there. It's not really a problem for bacteria because they have circular chromosomes in there with no ends, so they don't have to worry about it shortening. But what happens with eukaryotes is you've got this region on the end in here that they call it a telomere, that's kind of a buffer, so that you start reading beforehand so you can actually repair this. So each time you replicate, you've got a little bit that doesn't get repaired, that keeps getting chopped off. So each time. And eventually, it all gets chopped off in there. So this is your telomere in there that is going to be kind of that buffer sequence in there. And it's just made up of short 6 nucleotide sequences in there that postpones the erosion of genes. Because once you get past here, now you're going to start eroding the gene if you continue to replicate. So the telomeres is one of the theories of aging, is that you've got this amount of DNA that gets to be eroded before you start causing damage in there. And the older you get, the more of that is gone, and you're closer to eroding in there. So each human telomere is going to repeat TTAGGG between 100 and 1,000 times in there. So it will allow that cell to repeat between, or divide between 100 and 1,000 times in there, or that DNA to be copied between 100 and 1,000 times in there. When you have staggered DNA where you start to cut that off, it can lead to cell cycle arrest or death in there. So at that point, you can't replicate it anymore, and it dies. So telomerase is an enzyme that catalyzes lengthening the telomeres. And this is in eukaryotic gene cells, and it will help to restore their length. Most human somatic cells do not have active telomerase in there. And so having the telomeres shortened may be a way to protect you from cancer in there. That cancer can only grow so much and then it stops. However, a lot of the cancerous somatic cells have been found to have telomerase in there that allows them to continue growing in there. So last time, or a couple times ago, we talked about those the HeLa cells in there. They were the immortal cells that just could keep dividing and dividing and dividing. This is what allows those HeLa cells to keep dividing in there. Oh, we're almost done. Okay. So chromosomes consist of a DNA molecule that's packed together with proteins. You don't have a nucleus in the bacteria where it sits, but you do have a nucleoid region, which is just going to be that dense region of DNA in there. Chromatin is the complex of the DNA in there, in the nucleus. You're going to have these histone proteins in there that a good portion of them are positively charged with lysine or arginine on there to bind tightly to the negative charged DNA. It's the first level of packaging in there. So we've got the 4 most common types of histones are H2A, H2B, H3, and H4. So basically you've got these histones in here, and the DNA wraps itself around the histones. And then you've got another bead of histone that it wraps around like that. So the nucleosome is the bead of DNA wound 2 times around the molecules of each of the 4 main histone types. So histone, DNA together makes nucleosome in there. Yeah. Well, the bead with the DNA around it is the nucleosome. The protein without the DNA on there is the histone. So the linker DNA is the DNA strings between the beads. So linker DNA in between in there. So the histone tails in there of one nucleosome, the linker DNA, and the histones interact to coil into a 30 nanometer chromatin fiber. So this coils up into make this fiber that's 30 nanometers in there. And these get attached to scaffolding proteins in there to make these loop domains in there, and eventually form a 300 nanometer fiber. So you take coils and you coil them up again, and then you take what was coiled up again and coil it up some more in there. So it would be kind of like if you took your hair and you made lots of little braids, and then you started braiding the little braids, and then you started braiding those medium braids in there. It's just twisting on top of twisting. Yeah. It won't die that easily. Thank you. But I guess we might need to get a real mouse pad now. We're not worried about the water on the floor, if that's okay. It's just going to be a clean spot. I'm serious. They don't mop. Thank you. So the metaphase chromosome is those loop domain coils folded and further compacted into the chromosomes in there. So they end up with a width of 700 nanometers. And after all of this folding, we still end up with the genes always in the same place. So this folding's not completely understood. There is apparently some method to the madness in there that you get the genes in the same place every time you fold it in there. So heterochromatin is the interphase chromatin. It's the irregular clumps of it with, you can see them under a light microscope in there, but it's somewhat, it's fairly compacted in there. It's kind of when you start making the X's. The euchromatin is less compact, it's more dispersed in there. So when it's less compacted, the enzymes can actually get in there and reach the DNA. But when you fold it all up into chromosomes, they can't get in there because you have all of these loops and coils. So that's the end of chapter 16.